Let's see.
I don't know why, but I thought mine was. I thought it was.
What did one cat say to the other cat on Valentine's Day? You're perfect. Do skunks celebrate Valentine's Day? Of course, they're very sentimental.
Wait, hold on.
Skunks. Skunks. Sentimental. Skunks. Sentimental. Skunks. Skunks. Let's see what else was there. What did one blueberry say to the other blueberry on Valentine's Day? I love you very much. What did one light bulb say to the other light bulb on Valentine's Day? No, I love you very watts. Very, no, how did it go?
I love you.
Watts. It's watts. I love you very. I love you watts. I love you watts. 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 Yeah, I don't know. Okay, there's too many. There's too many of them. All right. So last time we have to finish we have to go over the answers to section 2 questions. We're gonna finish chapter 14 today. And then Thursday y'all have an exam. So I did want to show y'all this. So a couple of people have asked me, like, can you move back the exam? But what's on Tuesday?
Speaking of the case study, we're definitely going to move up or back the case study.
I was gonna say, can we switch the case study and the exam?
Wait, what?
Can we do the case study and the exam switch it?
I've never been more stressed.
No, it
About
actually
this
helps.
exam?
Well, no, because we have another instrumental
Okay,
exam
fine.
right after.
No, don't do
The
that.
name of what enzyme is literally like 5 syllables. Cool.
Wait, what?
One enzyme.
For a case study too? All right. Well, what about if we do this and do
Yeah.
this?
Yeah.
Yes, yes,
What about that?
yes,
Would that be okay?
yes.
This exam will be on Tuesday.
This exam would be on Tuesday, but I don't want to move the case study up because I don't think that's fair.
Yeah.
I mean,
I get your thought process.
you understand?
Even if everybody agreed, it just wouldn't work.
Yeah, that
Yes.
work?
Yes,
All right, I'll do that.
yes,
I did study for
Yes.
instrumental now.
yes.
About scale instrumental.
Okay, but y'all have to be on board on chapter 15.
Wait, what?
Like,
What?
we moved the exam back, so y'all have the weekend to study.
So we
Okay.
can study for
But y'all focus. You have to be like prepared for 15. Like you have to pay attention, because then we're not going to talk about 15 again. The 15 is going to be hard if we do this, because it's going to be a Friday, not a Friday, a Thursday, a Thursday, a Thursday. It's going to be big gaps. So I want you to realize that and work with that.
You don't understand.
Okay. Yes.
alpha 1, 3, and a transferase.
It's great. I love it. It's beautiful to my ears.
You just
No.
I told my mom never named that to be punished.
No, but it tells exactly what it does. It's so descriptive. I love it.
It's
Like the other part of that is saying
cost.
hydrogen bonding, and it's not true.
It's
right?
descriptive, but like at what
Okay,
cost?
so make sure you look and you know what's happening. Okay? All right, so take out your answers. Take out your answers to section 2. Yeah. I did, and did I tell y'all about my lovely dog? Did I tell y'all about my lovely dog?
Oh, pulling up the router line?
She pulled out one of the lines to my internet on last Thursday. And the guy comes out on Saturday and he's like, maybe she bumped it, like she wasn't bad, it's okay.
She did it again.
I came home yesterday.
She did it again, didn't she?
She had ripped the pipe that I had covered it with so that she wouldn't mess with it. The PVC pipe that I had embedded in the ground all the way up to the box. She had ripped it off. She had ripped out, the wire was ripped in the ground.
So you have no internet.
So I again have no internet. I love my dog. I love my dog. Okay. So you have your questions. Anybody need the questions? No, I don't think I've logged into iClicker. I think I've pulled it up, but I didn't start class. Thank you. I appreciate it. Did it show up? Yeah, okay. So, do iClicker. Thank you. Everybody got the questions? All right, so you should have answered section 2, right? So tell me, what's the metabolic logic for a reciprocal regulation of liver phosphofructokinase 1 and fructose 1,6 bisphosphatase activity by AMP and insulin? What you got? So let's talk about high levels of AMP first, right? Let's take them in sections. It's a little bit easier that way. What happens if you have lots of AMP? What does that tell you about the cell?
It's breaking down the sugar.
That you have a very low energy charge, right? So what enzymes are we going to regulate with high levels of AMP? There are 2. One of them is PFK1,
And the other is PFPase1.
FBPase1.
FBPase.
Okay, one goes up, one goes down. Which one goes up? PFK1 goes up, FBPase1 goes down. Why? Because an increase in PFK1 will result in
The glycolytic.
glycolysis. Glycolysis, yes, and glycolysis is going to end up increasing our ATP. If I could fit this, increase ATP, right? That's the name of the game. If we have a low energy charge, we want to have a more ATP, a higher energy charge. Okay, so what does downregulating FBPase1 do?
Turn down.
So that shuts off or downregulates gluconeogenesis, right? Because that uses energy. If we upregulated this, we'd have more and more and more and more AMP. And that would not be good. Okay, so that's AMP. What else do we have to worry about in this question? They asked about AMP and they asked about insulin. Okay, I want y'all to stop for just a second because we're going to talk about this today too in terms of glycogen. Insulin is a what? It's a hormone. Is AMP a hormone?
No.
How quickly do insulin levels change versus AMP levels?
Faster.
What's faster? Hormone changes are slower but longer lasting.
Oh.
Yeah, you have to get to the right organ, you have to release the hormone has to get to the target, right? So this is a long-term solution. This is a short-term. So this regulation is typically allosteric regulation, right? Binds to something, inhibits something, activates something, right? Whereas this is going to activate typically kinases and phosphorylases and things like that, right? And this is going to have a longer term effect, right? Because you need to be able to respond when you have a muscle cell that runs out of energy. How quickly do you need to respond to that? Right now. But how long do you go between meals? Hours, right? So that's how that's the difference between the two. So kind of keep that in mind as we're doing this. Okay, so we have high levels of insulin. What are we going to upregulate? Protein phosphatase 1. That is correct. Where are we for protein phosphatase 1? Like, we're going to give this descriptor, and we've really talked about 2 different places in the body. This is the liver. We're really talking about the liver here, okay? So what's going to happen if we activate protein phosphatase 1? What are we going to do? We're going to desphosphorylate that dual enzyme of PFK. This time it's not PFK1, it's PFK2 and fructose 2,6 Bisphosphatase, right? Now, if we dephosphorylate, is that going to activate the kinase activity or deactivate the kinase activity?
Deactivate.
So it activates it. It activates the kinase activity. So then what's the like the physiological, you know, okay, so this kinase is now active. What is it going to do? What is it going to phosphorylate? Fructose 6 phosphate 2 6 because it's 2 6. So if we have fructose 2 6 bisphosphate hanging around,
It's downregulated.
yes, okay, so this is then going to increase glycolysis, GLY, glycolysis, and also Whew. How you feel about that? Good. Good. Apparently now you have the weekend. Okay. All right, so that was the first one. Did that answer everything? Yes. Okay, so number 2, right? We make lactate in the muscle cells. But if lactate builds up, what do you have? Muscle cramps, right? So then we have to transport the lactate somehow to get to the liver so that we can turn it into glucose, right? Okay, so the question is, we have the Cori cycle. Involves anaerobic glycolysis and gluconeogenesis in livers to convert glucose to lactate back to glucose. Explain why the Cori cycle has a net cost of 4 moles of ATP. So that's the first one. Why does it cost 4 ATP? Moles, molecules, whatever. Why does it cost 4 ATP to go through the Cori cycle? Yep. Gluconeogenesis. I always have to like think about it as I spell it. Cost, yes, yes. Cost 6 ATP. But glycolysis produces, now this is all net, right? Produces 2. So the net cost is 4.
The answer was that it costs 4 ATP. That's actually ATP, but it was because the answer was 6. So like, yes, I know what you're talking about.
So these are because it was GTP,
Yeah, but we say 6 ATP,
but it's technically 4 and 2. But GTP, assume GTP. I'm going to make this statement. Assume GTP is ATP. Okay. For all mathematical discussion, assume GTP is ATP. Sometimes they'll say ATP equivalents. Sometimes they'll say ATP. As long as you understand that's where the 6 comes from, both ATP and GTP. Okay, but then there was like multi-part, right? Okay, let's see. What might be the evolutionary advantage of the Cori cycle? Like think animals, think primal prey running from predators. Well, so if you keep running, Cori cycle's not going to work. You have to like slow down, right? Cori cycle, you need a period of rest. So if you can run long enough to get a break to climb up a tree, right? Give your body a rest. then your muscles will have a chance to build that ATP back up. And then, okay, I can sprint again. Right? So that's kind of the evolutionary. You gotta think that we've been evolving for a very long time without technology that has made our lives easier and that we don't have to run from predators anymore, right? So our body is still designed for that predator-prey relationship, but we don't use it anymore.
Adapt to the environment, we literally adapt to the environment to us. So
All right, and then let's see. What is an everyday biochemistry example of how the Cori cycle is utilized by athletes who run or swim and sprint in races, right? So what do they do? What do runners and swimmers?
They like slow down, but they don't stop.
Yeah, they don't stop. Like you don't see them get out of the pool lickety split. You don't see sprinters just drop at the finish line, right? They keep walk it off, right? You've heard that, right? Keep walking, keep moving.
That's why there's so much paddling when they're like sitting at the neck. It's slow and effective.
Yep, they just doggy paddle, they just move.
we have to cycle
Yeah.
all that.
That lactate, yep.
Lactate out of it.
Okay. So let's see if we start. All right. So good. Go ahead.
Can we read? I'm sorry. Can you explain the evolutionary advantage of the course?
So it's the same thing. It's the same thing as the runners, right? If you are running, eventually you are going to run out of ATP. You're going to collapse and get eaten.
So the
Right,
course
you
cycle's
will.
advantage is to just build back up the ATP.
Is that if you get a rest, you can build back up the ATP that was in your muscles. Otherwise, eventually you're going to quit. Is in the rest period. Yeah. Okay, good. All right. So now glycogen. Ooh, I love glycogen. It's a strange thing to say, right? Okay. So remind me about glycogen. We talked about glycogen is not just a straight chain, right? It is branched. So what kind of connections? They're showing us we have branches at an alpha 1,6, but what's our chain linkages? So you have alpha 1,4 change, right? And then you have alpha 1,6 branches. So how many anomeric carbons are participating in the glycosidic bond? Just look at the designation. If your anomeric carbons participating in the glycosidic bond, what do you have to do? Name it as alpha or beta. So if I only see one alpha,
It's not, it's only one element.
that's only one anomeric carbon, right? Okay, so you gotta keep remembering those things that we've learned in the past, right? Okay, so I think this is the no, this is good. Okay, could remember. So remember, glycogen is like that cush ball, right? It's got a center, and then it's got lots of little fingers that are going outwards. So tell me about the difference between the center of the cush ball and the outer fingers of the cush ball. Tell me about sugars. Remember sugars, reducing and non-reducing ends. Remember that? So the center is the reducing end. The ends are the non-reducing ends. Very good. So if you have a branching mechanism, right? The more fingers you have, the more non-reducing ends you have. The more non-reducing ends you have, the more substrates you have for processing of glycogen into glucose. And that means the quicker you can get glucose flowing in the bloodstream. And why is that evolutionarily advantageous? Because if a bear walks in the classroom, right, we want blood sugar to spike so that we can run, our eyes will dilate, our breathing rate will go up, our heart rate will go up, right? All this kind of stuff that you need energy for, right? And I'll be the slowest one, so y'all go ahead. It's cool.
Not to get that.
So-
I mean, I get it, but I think we, like, in the
I-
wild,
I-
some bears.
I think you just bring a gun.
Yeah, but we can't do that here.
No, there's not gonna be a bear come in the classroom. If you're going camping, you know, that kind of stuff, you know, definitely protect yourself, but okay, so, we have branching, and this branching doesn't occur perfectly at the same place every time, but we know it's always gonna occur about every 8 to 12 residues, which is more frequent than those storage molecules in plants, right? Because plants don't have to run. So when you talk about this, there's got to be something in the center to start this, right? So you actually have a protein core of glycogen that is at the center of the Kush ball. So you have a protein there that's kind of dual function. It actually serves to bind that very first sugar molecule, and it catalyzes the first couple of addition reactions, and we'll talk about it. But the whole point is that once we have this highly branched glycogen molecule, we can generate glucose 6-phosphate like that, right? So if you look, depending on where you are, if you're in the muscles, about 1 to 2% of muscle mass is glycogen. But if you look at the liver is about 10% glycogen, which is quite a bit. Muscle just stores for itself, right? It's not altruistic, right? Self-centered. But the liver, who's the liver mostly worried about feeding?
The brain.
The brain, right? Very, very important. Very, very important. Okay. So we're going to talk about two parts of glycogen. We'll talk about first degradation, and then we're going to talk about synthesis. So all of this is going to occur in the cytosol of the cells. And we have 3 big key players in this: glycogen, phosphorylase. So, and then glycogen synthase. These are kind of opposites of one another. So what's a common way to regulate enzymatic activity? Allosteric and? Well, yeah, that's true. But allosteric is through energy charge. Yeah, post-translational modification,
Oh,
right? Phosphorylate, dephosphorylate. So just like what we talked about before, phosphorylation is going to be a huge thing here. So remember, glycogen phosphorylase and glycogen synthase are going to have opposite effects on glycogen. Therefore, if we phosphorylate it, let's say we add a phosphate group, right? This is my abbreviation for you phosphorylated the enzyme. Once you phosphorylate that enzyme, we want different things to happen. So if we have glycogen phosphorylase, we are going to activate the enzyme. If we have glycogen synthase, we're going to phosphorylate, do the same post-translational modification, and guess what? It's going to inhibit its activity. Right? And that's exactly what we want. Because if you just activate kinase activity, now we can have opposing effects on opposing pathways, which is what we really, really want. And then we'll talk about branching and debranching, but they're kind of a different ball of wax. Okay. So, all right. So remember, we have in glycogen a chain, and then we have a branch. So if we look, here's glycogenin, here's our protein, right? And we're linked to our very first glucose molecule via an O-link, right? So if you look at this molecule, this is our straight chain. So what's our, I wish I could zoom in. What's our linkage here? This is my alpha 1,4, okay? What is this linkage here? This is my alpha 1,6, okay? So if you look at first glycogen phosphorylase, what is glycogen phosphorylase doing? we have this straight chain. This straight chain has a branch. In the presence of glycogen phosphorylase, what happens to this last residue, this blue glucose moiety on the end of glycogen? So we are going to release one molecule of glucose from the non-reducing end of glycogen, right? Okay, so in order to do that, we do need to have an inorganic phosphate present because we're going to release it as glucose 1-phosphate, right? Now, this is going to be really important what state the glucose is in so that we know what we can do with it. Because if we want to export it out, right, we're going to convert it to glucose 6-phosphate. Now we have a substrate that can do one of two things. Either if it's in muscle cells, we can use it in glycolysis, or we can dephosphorylate it and then ship it out, right? So it depends on which place you are, what you want to do with it. But regardless, you need it in the glucose 6-phosphate conformation, right? Okay. So when would you want glycogen phosphorylase to be active? When you have low blood glucose. Try not to write BS. Blood sugar, right? Blood glucose. We're gonna stick with that. So if we have low blood glucose, right? We haven't eaten in a while. So we're going to trigger what hormone release? Glucagon. The other thing is maybe we don't have low blood glucose, but the bear just walked into the classroom, right? What hormone is going to be released then? Epinephrine, right? That's the fight or flight. Okay, so that's glycogen phosphorylase. Now let's kind of flip the coin and look at glycogen synthase. Glycogen synthase is going to be the enzyme that adds a glucose to our growing chain. Where is it going to add? To the non-reducing end, because the reducing end is bound to our protein core, right? Okay, so the trick is that you can't just add a regular old glucose molecule to our growing chain. What you actually have to do is you have to start out, right? You've gotten glucose, you've eaten. Glucose is in the blood, you secrete what hormone? Insulin, right? Okay, so now you could import glucose into the cell. You have hexokinase. Hexokinase will phosphorylate the glucose. It's trapped in the cell. Once it's trapped in the cell, then we convert it from glucose 6-phosphate to what? Glucose 1-phosphate. Now, in the presence of UDP glucose pyrophosphorylase, woo, I can speak today, we are going to actually attach it to a UTP molecule. And when you attach it to a UTP molecule, right, you're going to release 2 inorganic phosphates, and you're going to end up with UDP. So you have 2 phosphates still attached, and then 2 released freely to this nucleotide, right? Now, this is a substrate for our enzyme glycogen synthase. So it has to be, the glucose has to be processed in order to attach it to our growing glycogen chain. This is the substrate. This is the substrate, right? That's required for glycogen synthase, right? You can't just have glucose come in and hook it onto the chain. The point is it has to be processed in order to be the correct substrate for glycogen synthase. That's what you were looking for? Good? All right. Okay. So that's our big 2, right? That are opposing one another. Yes. It does not attach free glucose. Oh yeah. It attaches just glucose. And we're going to look at that mechanism in detail. But yes. Okay, so we did our opposing glycogen phosphorylase and glycogen synthase, right? So now you can see if you upregulate one, what are you going to want to do? Downregulate the other, right? And vice versa. You're always gonna have one up, one down. So that, remember that seesaw that I told you that was super important? Same thing here. Okay, but we have a third group of enzymes, right? Our glycogen branching and debranching enzymes. Yeah.
Why is it the two, why is it the PPI?
Because this is pyrophosphate, it's two inorganic phosphates. And I'm going to talk about that. I'm going to talk about why that's important. That's super important. I'm going to talk about it. Okay. So we have our debranching and our branching enzymes. So these are the ones that are going to give us that maximum number of non-reducing ends that we really need in order to have a rush of glucose into the body, into the bloodstream, right? So this is always going to cleave our branched alpha-1,6, right? That's why we wrote it here. The other one, which is glycogen phosphorylase, is going to cleave the alpha-1,4, right? So you have two different enzymes that can cleave glycosidic bonds. You have glycogen phosphorylase is going to cleave alpha-1,4. Our debranching is going to cleave alpha-1,6. Okay. Yeah. And then and then you have enzymes that can do the reverse, that can actually create a branch. And we'll talk about how they do that. Yeah. There's a lot on this slide. I'm going to show you. We got to get more into detail before we talk about that. Okay. So when your muscles contract, right? Typically, in animals, don't think really about humans, think about other animals, right? When animals are running, when animals are doing things like that, right? The primary source of ATP production becomes glycogen degradation. That's why muscles always have a store of it, because that store can last for like an hour, right? Of continuous muscle contraction, maybe not extreme, but just continuous muscle contraction, about an hour. So that's a really long time. So we initiate this by glycogen phosphorylase, right? This is our enzyme. And this enzyme exists in two conformations. Remember hemoglobin, when we did the R state and the T state? It's the same sort of deal. The R state is the active state. The T state is the inactive state. And there are two different ways that we can regulate active state and inactive state. One of them is post-translational modifications, right? Phosphorylation, dephosphorylation. And the other is by allosteric regulation. And we'll talk about both of those too. So if glycogen phosphorylase is active, it is going to catalyze cleavage of our alpha-1,4 glycosidic bond, but only at those non-reducing ends. That's why we need the branching, so that we have lots of non-reducing ends. It's going to keep cleaving until it reaches a point where the molecule is 4 glucose units from a branch point, and then it can't work anymore because it's no longer in the correct conformation. So it basically stays attached and it ratchets along, right? But when it gets to when it gets to a branch point, it can't go anymore. So let's look at how it's regulated, right? I'm going to say that we're secreting glucagon, right? If we're secreting glucagon, tell me what we want to do. What's our end goal? Raise our blood glucose levels, so we need to degrade glycogen. So if you kind of think about, I want you to, when you're like studying this, when you're thinking about it, I want you to think big picture, then details, then big picture again. It's going to help it make more sense to you. Okay, so if we have glucagon or epinephrine, either way, right, we want to metabolize the glycogen that we have stored. So that is going to activate phosphorylase kinase. Phosphorylase kinase, it's a kinase. So it is going to add phosphate groups to, here's our glycogen phosphorylase. It's in the inactive form. It is going to phosphorylate it. When it becomes phosphorylated, now do you see these little stars? This is the book's way to say that this is an active enzyme. So this is active glycogen phosphorylase. So this is active glycogen phosphorylase. This is inactive. And I kind of got to the end there, but y'all know what I mean. Okay. So if you have glucagon and epinephrine, basically you're going to start triggering a phosphorylation cascade, right? So we have phosphorylase kinase that becomes active. Phosphorylates are glycogen phosphorylase. Now, the opposite of true is if your body is secreting insulin. If your body is secreting insulin, what do we have? High blood sugar, right? High blood glucose levels. So now, if we have high blood glucose, we're going to activate a protein phosphatase. What does a phosphatase do? Removes our phosphate. So it takes these active glycogen phosphorylase molecules, cleaves off our phosphate groups, turns them into inactive T state, right? So you have one enzyme, right, that's controlled by two different hormones just through phosphorylation or dephosphorylation. Okay, all right. What's that? This is covalent modification. This is phosphorylation is covalent modification, right? Because you're creating a bond or you're breaking a bond. Either way, make or break a bond, you're covalent. So if you have active glycogen phosphorylase, what's going to happen? Oh, I want to be in blue. Glycogen. What did you say? Degradation. Okay, how about if glycogen phosphorylase is dephosphorylated and it's in the T state? We're going to inhibit, right, glycogen degradation. So that's an inhibitory, right? You could write over here, you will increase. Here you're going to inhibit. Telling you, explain this to somebody else, even if they have no idea what you're talking about. It's okay. All good. They're gonna think you're so smart because y'all are. Y'all are. All right. So now we're going to talk about glycogen phosphorylase kinase. So there are really two ways, right, that we said to regulate glycogen phosphorylase. You can regulate through phosphorylation or you can regulate through allosteric. Okay. So let's look. We're in two different places, right? We're in muscle cells versus liver cells. So let's look at our muscle cells first. We said that we can have an inactive T state, and in the presence of epinephrine, we activate phosphorylase kinase, right? Yes? And it gets phosphorylated. Now, what state is it in? If it's phosphorylated, it's in the active state, right? That's what we kind of already talked about. Okay, well, what happens if you have molecules that are in the inactive state? You have not secreted epinephrine yet, but you have this allosteric effector site. An allosteric effector site is a place on the molecule where you have a regulatory molecule that will bind, change the conformation of the enzyme, and therefore regulate its activity, right? We learned that in Biochem 1. Yes? Okay. So now, in the presence of, let's say, AMP, right? My cell has been working hard. It has run out of ATP. What happens to levels of AMP? AMP goes up. If AMP goes up enough, and AMP binds to our inactive, our dephosphorylated phosphorylase kinase, not phosphorylase, glycogen phosphorylase, right? It is going to become active even though it's not phosphorylated. So we have two ways to activate it. You can activate it through a hormone that will phosphorylate it, or you can activate it through energy charge of the cell, right? This is our post-translational modification. This is our allosteric control. So in low levels of ATP, AMP binds and activates our enzyme. And now we can go in and do what? Whether you're in the active state for either one, you're going to degrade glycogen. Now, here's the cool thing. If you have something that is in the T state, right, and it binds AMP, it actually shifts it to look more like the R state and is therefore more likely to be phosphorylated. If you have our glycogen phosphorylase that is bound to AMP, it mimics the active R state and is therefore more likely to be phosphorylated.
Oh, almost like a two-step instead of a one-step.
Yep. Yep. So you're like primed to be phosphorylated. So that you can have hormonal control and you can have energy charge control. Right? Yeah. Now what happens if you've been secreting epinephrine, so you're phosphorylated, so you're active, but your cells stop working as hard? What happens to the energy charge of the cell? It goes up. Energy charge goes up. So as I'm making more ATP, what else is accumulating in the cell? Glucose. Guess what else binds at that same site? Glucose. Glucose will promote the T state. The T state is the inactive state. Right? So if you have lots of glucose around, you want to degrade glycogen. No. So the two allosteric control mechanisms are AMP and glucose. Yeah. Okay. So now let's switch a little bit and let's talk about liver cells, right? Okay. What do you see as a difference between liver cells in general and muscle cells? Look at the two diagrams.
The liver cell has two T states instead of two R states.
What is not binding to the allosteric site? AMP.
We're not wrong though.
So in what cell type is energy charge more relevant and more necessary for control? Muscle cell or liver cell? Muscle cell, right? Because muscle cells change energy charge like that, right? The more they work, the more they're metabolizing that ATP, so they need to regain it quickly. Liver cells is a much longer term, so they're more dependent upon hormonal regulation than they are allosteric regulation. Does that make sense? But they are still regulated by glucose. Yeah. You're not wrong. You were not. Did I say that?
No, you just looked at me like really.
No, I was enjoying it all. I was really enjoying it all. They were all true statements. Y'all remind me very much of my son when y'all do that. Because, well, no, I really like it. He's very meticulous when it suits him. If you know what I mean. You know, if you give him directions and he's like, well, you said. I'm like, oh, I did. So it keeps me in check. It keeps me in check. I like it. Okay. So
I've witnessed this firsthand.
you have witnessed it. Yes, you have witnessed it firsthand. All right. So when we talk about glycogen phosphorylase, enzymatically, if it is active, what does it do? breaks down glycogen, right? It releases glycogen. So what's going to happen is glycogen phosphorylase does not act alone. It acts together with a coenzyme called PLP. So PLP, so you see the enzyme, you see how much emphasis we put on the enzyme? We just put ENZ. You like that? So this enzyme has a phosphate group attached to it, right? So do you remember how I said this reaction requires a free inorganic phosphate? So the phosphate on the PLP is actually going to have a covalent reaction with the inorganic phosphate. Now the oxygen on the inorganic phosphate is going to attack the glycosidic bond. So we're going to break the glycosidic bond there, and we're going to release a free glucose, but that free glucose, right, because we attacked the glycosidic bond, now has a phosphate group attached to it. So that is where the first phosphorylation of that glucose comes from. It actually comes from the removal. So it's a nucleophilic attack, right? The PI, the one PI that it needs. So then basically what it's going to do is the enzyme is going to slide down the chain. It removed the blue glucose molecule, right? So the next molecule is the green one. And it's going to repeat exactly the same thing with a new inorganic phosphate. And it's going to attack the glycosidic bond between the green sugar and then the white one, the next one, right? And it's going to keep going and keep going and keep going until it gets too close to a branch point. And that too close is within 4 of a branch. So what is your product? You get glucose 1-phosphate because this is an alpha 1,4 linkage, right? So you're attacking here the carbon, that's the anomeric carbon that's normally participating in the glycosidic bond. That's why you gave it an alpha, right? So now we have that phosphate, and you see the alpha orientation, right? One up, down, right? Okay. All right. So now we also need to talk about, you don't just have a straight chain, you also have branches. So what happens when you get to a branch point? So you have a debranching enzyme. The debranching enzyme recognizes that you have a partially degraded branch, right? Your branch, you have basically a short branch now. So what it's going to do is it's going to transfer, because how far away are we from the branch point? 4 units, right? So what this debranching enzyme is going to do is it's going to actually take 3 glucose molecules from that branch and add them to the nearest non-reducing end. Now what do I have left? Just 1 glucose monomer attached via an alpha 1,6 linkage, right? So now that enzyme can cleave that 1 single glucose molecule and release it. Okay, so here's the enzymatic process, right? So you get to glycogen phosphorylase, right? It cuts. Now we are 4 away from a branch point. So our debranching enzyme is going to take these 3 and it's going to hook them on to the nearest non-reducing end, so the nearest little finger end. And we're going to be left with 1 glucose molecule on an alpha 1,6 branch. Now they're showing it's not that far away really, but sometimes it's much farther. So now you have that 1 in that alpha 1,6 linkage. You have your same debranching enzyme. So your debranching enzyme has to have multiple activities. One is a transferase activity, and one is a glucosidase activity, where it's actually breaking the bonds. The glucosidase activity is in the second step, where we take away this last sugar that's attached in the branch and we release it as a free glucose molecule. And now we have a longer chain, and this longer chain can be acted on by glycogen phosphorylase. So here's the difference. Look at what's released. how are the products different from our glycogen phosphorylase versus our debranching enzyme? Here we have straight glucose. Here we have glucose 1-phosphate. So you actually do end up with a different product. But most, like 90% of the degraded glycogen comes from glycogen phosphorylase. Thank you. My brain's skipping ahead to synthesis. Cool? All right. So now we're going to change gears, right? Now we're going to talk about, we have a high energy charge. What do we want to do? Make glycogen, right? Name of the game is make glycogen. So how are we going to get glucose into the cell? We're going to have free glucose. What's the very first reaction we do to free glucose? Phosphorylate it. We phosphorylate it because we need to trap it inside the cell. Okay, so we phosphorylate it. Now we go through a mutase. So we're going to change that phosphate group from being glucose 6-phosphate to being glucose 1-phosphate. So you remember how I said you're going to attach this glucose to a UTP, not an ATP, but a UTP. It's got 3 phosphates attached to it, right? Triphosphate. So this alpha phosphate, the one closest to the sugar, this is the one that undergoes an attack from the phosphate that's on the glucose 1-phosphate, right? And so what you end up doing is breaking the bond between that alpha phosphate and the next 2. So what you release is a pyrophosphate molecule, which is 2 phosphate groups linked together, and you end up with UDP glucose, which means you are left with 2 phosphate groups attached to the sugar, or attached to both sugars, right? Okay, so why does this work? This seems like a lot of work to do in order to just attach it to glycogen. How can you drive this reaction forward? What do you need? You need a low delta G in what kind of a reaction? No, an intermediate. Remember we talked about intermediates, right? That you had to have multiple reactions and that the intermediates canceled out, but yeah. So if you look at this pyrophosphate does not like being in this conformation. It's very unstable. So very quickly, it is going to be cleaved and we're going to get 2 inorganic phosphates. This has a very large negative delta G value. The others are basically 0, right? Remember, we've talked about that. Basically 0 means you can go back and forth. But what happens when you have a really high negative delta G? You've committed, you're done, right? So this is what drives the creation of UDP glucose. You need something to drive it. You need something to make it beneficial for the cell, and that's what this is. Isn't that kind of cool? All right. So now we got to kind of put them together, right? Okay. So if we start out, right, we have our glucose 1-phosphate. We're going to go through our reaction with UTP, and we're going to create UDP glucose. It's this UDP glucose that is the substrate for glycogen synthase. Glycogen synthase cannot work with glucose 1-phosphate. It can't. It needs the UDP glucose. The UDP glucose is then processed by glycogen synthase, right? We cleave off the nucleotide portion and we attach the sugar to our growing chain, and we're going to always attach to the non-reducing end, okay? And then we have a new substrate and we can just keep reusing it, right? So it's a very cyclic but useful process, right? Okay, now you're going to have glycogen synthase. It's going to keep making this really long chain, right? But we know that we need branching in order to make this glycogen really suitable for the cell, right? We need lots of branching so that we can up our blood sugar really quickly. So how do we do that? You have to start with a really long chain. Then you're going to have a branching enzyme that is going to go in and actually cleave a glycosidic bond within the long chain and then attach it to the carbon 6 molecule of a sugar moiety on another long chain. So you're going to create branching that way, right? You're not going to start a branch and then add from it, right? You're going to have to make a really long chain and then break off part of it and then stick it on the top of a previous long chain. Now, the enzyme required to make this branching happen is relatively large. So can the branch points be extremely close together? No. So the branch points, that's why the branch points are never less than 8 sugar residues apart from one another, right? It's usually 8 to 12. But you have to leave room for the enzyme to actually work. So that's one of the reasons why it's that far away. All right. So we've kind of talked about how to make glycogen, but everything we've talked about so far, we've assumed that there's a glycogen molecule there to begin with, right? So how do you start from scratch? You need a protein called glycogen in order to start that process. And glycogen has two basic activities, right? The first one is it's going to act as the anchor for that very first glucose molecule, right? It is going to form a glycosidic bond. What residue? It's tyrosine, right? It's written on there. It's tyrosine. Tyrosine has what? An OH, right? So now we know we can form an O bond with our glucose residue. So now we have one. But if you remember glycogen synthase, it needs a chain there in order to start rolling. There's not a chain. The enzyme doesn't fit. So this glycogen has to have additional enzymatic activity that can then extend, it's going to extend 7 glucose residues. Why 7 glucose residues? Remember? They're similar size in that you need 8 residues for your branching enzyme. You need about 7 to 8 residues for glycogen phosphorylase, right? In order for it to glycogen synthase, in order for it to go, right? So you're going to just keep adding and adding until you get to 7. Then glycogen synthase can take over. So it's a pretty cool. That's a pretty cool little core molecule, right? All right. So we're almost to the end. We talked a lot about post-translational modification, right? So we're going to talk a little bit more about hormonal regulation. So when you talk about epinephrine or glucagon, right? There's actually a variety of proteins inside the cell that are activated by epinephrine and glucagon. Do you remember when we were talking in, oh God, what chapter was that? I don't even remember. We were talking about all the receptors. Yeah, I don't remember. But you remember like your phosphorylation cascade, that you had multiple kinases that were activated, right? You remember, sort of remember all that? Well, the same thing's true here, right? Epinephrine can trigger multiple cascade pathways, but they're all kinases. You see how that's common throughout them all? They're all kinases. So if you look at glycogen synthase can be regulated by, again, mechanisms, right? One of them being post-translational modification. So we have an active state. This active state is the R state, right? It's going to go and make glycogen. But what happens in high levels of epinephrine and glucagon. Do you want to be making glycogen? No, I'm running from the bear that entered the classroom. I don't have energy or time to make glycogen. So what I'm going to do is I'm going to activate kinases. The kinases are going to phosphorylate our enzyme, turn it into the T state, and shut it down. If we shut it down, we stop glycogen synthesis or formation. Right? Insulin, on the other hand, activates what? Phosphatases. In general, phosphatases. Phosphatases will remove phosphate groups, right? Now we're going to convert our enzyme from being active. We're going to dephosphorylate it and make it, I'm sorry, it's inactive, it's phosphorylated. We're going to dephosphorylate it and make it active. So here we're going to make glycogen. All right, now ready to put it together? You got this, right? All right. So we got two opposing conditions, right? In the same cell. If we have low blood glucose, we're going to make glucagon. Glucagon is going to be recognized by what kind of a receptor is that? Look familiar? The G protein, 7 transmembrane, right? That's all the sneaking up and down. All right. If we have glucagon, we are going to activate kinases. So we're going to have a net reaction if we're going to phosphorylate stuff, right? If we phosphorylate stuff, we're going to initiate that cascade. The cascade is primarily controlled by protein kinase A. Protein kinase A goes in and activates phosphorylase kinase. Phosphorylase kinase is going to activate our glycogen phosphorylase, right? It's a phosphorylase kinase. So we're going to convert from inactive glycogen synthase to now having active glycogen phosphorylase. So we need both things to happen. We need to have active glycogen phosphorylase so we can break down glycogen, and we need to have inactive glycogen synthase so that we can not make glycogen. So if you downregulate one, you have to upregulate the other, and vice versa. Correct. You do not want fetal cycling. Net reaction of inhibiting glycogen synthase and activating glycogen phosphorylase is glycogen degradation, right? We degrade glycogen, we can pump glucose out. We can send it to the brain. Now, if this were a muscle, you'd stop right there, right? Right, saves it for itself. Okay, so opposing, you've just eaten, you have high blood glucose levels, so you secrete what hormone? Insulin. Say it again. Sure is. Amazing how you see these things again. So insulin induces our net dephosphorylation events. Net dephosphorylation events are controlled by protein phosphatase 1, right? See how dephosphorylation, phosphatase, phosphorylation is a kinase. So what two things do you want to happen? You want to shut down glycogen phosphorylase, and you want to activate glycogen synthase, right? So you just tip the seesaw the other way. Our net reaction is we're going to make glycogen. If we make glycogen, if our blood glucose was high, now we're importing glucose and we can use it in glycogen synthesis. Yeah. Try to draw that. That would be a really good, like, tie it together. Now this does not include what? This is all one kind of regulation. What kind of regulation is this? It's hormonal and therefore post-translational modification. So is there another form of regulation? Yes. allosteric energy charge regulation. So this doesn't quite tie it all together, but it's pretty close. Pretty close. All right. So there's one other way that y'all actually did talk about earlier today and how you regulate enzymes, and that is cellular location, right? So when you secrete insulin, it does many things, right? One of those things is that eventually it will activate the insulin stimulated protein kinase 1. And so that will activate what's called GM. GM is a helper protein that will actually take protein phosphatase 1 to the glycogen. So it will actively transport it to the correct place in the cell. So now that glycogen synthase is in the right place, right, then we can regulate what's going to happen with those enzymes. So in this case, right, if we have insulin, have we eaten or are we in starvation? We've eaten. So what do we want to activate? Glycogen synthase. What do we want to deactivate? The phosphorylase, right? So it's just another level of regulation. And you can have inhibitors for those transport molecules, all that kind of stuff. So as much as we kind of simplify this, there are many more levels of complexity. But all right. So here's kind of our epinephrine, right? That was insulin. So let's kind of talk real quick about epinephrine. Epinephrine, and this is going to be specifically in our muscle cells. So if you activate epinephrine, remember our signaling cascade, and you have those second messengers of cyclic AMP. So cyclic AMP, we learned, activates protein kinase A. This is those pathways that you were like, oh, why do we have to learn these pathways? So it's going to do two things. Protein kinase A is going to phosphorylate our recruiting protein, right? So if we phosphorylate the recruiting protein, we're no longer going to take glycogen synthase to the right place. So that's going to be a level of down-regulating glycogen synthesis, cellular location. All right. The other thing that protein kinase A will do is it will actually phosphorylate an inhibitor. This inhibitor will inhibit protein phosphatase 1. What does protein phosphatase 1 do? Right. So what we want to do is we want to inhibit protein phosphatase 1. So if you phosphorylate the inhibitor binds to protein phosphatase 1, we end up having a downregulation of glycogen synthesis, right? At the same time, we're going to promote glycogen degradation because epinephrine activates cyclic AMP, which activates protein kinase A. Well, it's a kinase. It's going to phosphorylate our two enzymes. When we phosphorylate glycogen phosphatase, we're going to break down glycogen. When we phosphorylate the synthase, we're going to turn it off. And now we're bringing it all together. Glad y'all have the weekend. Glad y'all have the weekend. All right. There are so many different steps and so many different enzymes that are crucial to making all this happen. Wow, we went pretty over, but that's okay. It was good to spend this amount of time on this. That if you mess up any of the enzymes involved in these pathways, you are going to end up with a glycogen storage disease. And like I could teach a class on glycogen storage diseases. Depending on where the enzymatic deficiency is, it gives you different symptoms. They're relatively rare disorders, but if you go to med school, you're going to have to learn them. But we don't have time to do that. So lots of different disorders. What? You don't have to learn them now. There's only so much. Like if you understand the mechanism of how it works really well now, Then when you get to med school and you're starting to learn the individual disorders, you're gonna be like, oh, I know what that enzyme does. So then it makes sense with the problems. So, well, yeah, in my spare time. In my spare time. Okay, so we have 2 questions left on our little handout, right? How much time we have? 10 minutes? Or 10 minutes? All right, so I'll give y'all 5 minutes and we'll go over the answers. Even if you're not to the end, at least I want you to go home with the answers so that you could study. Yeah, you could turn, you can talk. I'm gonna pause the recording.